Hello and welcome to my video on protein digestion and protein metabolism. Before I jump into digestion and metabolism, just wanted to make a brief note on what is protein. And protein is made up of amino acids, which are the monomer form of proteins, and are combined into longer chains and structures, which are referred to as the polymer of protein. To form certain structures within our body. And these different structures are the primary structure, which is just the amino acids linked in a chain to form a polypeptide. The secondary structure, which are your alpha helices and beta sheets, and then your tertiary structure and quaternary structure. And these different structures make up different things within our body. So your muscles, your neurotransmitter, hormones, enzymes for reactions, and different antibodies within our body. When we ingest food, our body breaks the protein from its polymer form back into its monomer form into these amino acids, where then our body will send it to the liver, where the liver will decide its fate. When we have two different kinds of amino acids. We have essential amino acids and we have non-essential amino acids. Your essential amino acids are going to be the amino acids that your body cannot synthesize on its own. I mean, it can't make it on its own, so you have to get it from your diet. The non-essential amino acids your body can synthesize on its own. In addition to those, you have complete proteins, which come from animal products such as meat, poultry, eggs, and dairy products. And the complete proteins are proteins that have all the essential amino acids that you need from your diet. Incomplete proteins are things such as vegetables, tofus, peas, grains, nuts, and seeds. Those do not contain all the essential amino acids, and therefore you need to have complete protein in your diet as well. So now into digestion. So for this example, we're going to be talking about a steak. If you eat a steak, it's going to be sent into your stomach in a bolus form here. On the lining of your stomach, you have different cells. And these cells, your parietal cells, are going to secrete HCl. Your chief cells are going to secrete pepsinogen. And the combination of this, the HCl is going to activate the pepsinogen to become pepsin. The G cells are going to secrete gastrin, which are just going to enhance the secretion of these other HCl and pepsinogen from the P cells and G cells. And then as your ribeye steak makes its way into the duodenum, and what's now called, it's going from bolus and now it's going to be coming out of the stomach as chyme, your body is going to start secreting from the duodenum through cells in the duodenum. We'll enlarge it right here. Your eye cells are going to secrete CCK, which are going to go to your CNR cells in your pancreas to release the pancreatic juices, also known as zymogens. It will also release, the S cells will release secretin, which will go to the duct cells to release bicarb, which bicarb is going to neutralize the acidic environment coming from the stomach because of the HCl. Is going to neutralize the acidic environment the chyme that's coming from the stomach, and that is also going to activate enterokinase, which will further activate the zymogens, which I'll talk about right now. Once the zymogens and the bicarb are activated, the enterokinase activates trypsinogen, which is then going to be turned into trypsin. And trypsin is then going to activate procarboxypeptidase to become carboxypeptidase, and chymotrypsinogen to become chymotrypsin, to further break down the polymer form of amino acids into amino acids and di and tripeptides. Once your amino acids and di and tripeptides make it to the jejunum, they are going to be absorbed through the brush border, through the enterocyte, and into the blood to go to the liver. You have your amino acids that make it in through various different transporters, and then are facilitated, or through facilitated diffusion, are diffused across the basolateral membrane into the blood, into the liver. Your di and tripeptides are aminopeptidase and dipeptidase can act on these to break it down even further, but di and tripeptides are brought in through the PEP-T1 transporter with a hydrogen ion, and once in the cell, in the enterocyte, intracellular peptidase will act on it to break it down into amino acids, and then through facilitated diffusion go across the basolateral membrane into the liver. Once your amino acids are going to the liver, your liver is going to decide what to do with the amino acids. And there's two things that your liver can do with the amino acids. First, it can go through transamination, which in a healthy person you should be going through this process about 90% of the time. This is where basically your amino acids are going to be, your body's going to be adaptively recycling the amino acids into the biosynthesis of new nitrogen products, such as neurotransmitters, or into the nitrogen pool, which will be held by glutamate. So your amino acid is, the amine group of your amino acid is taken off through the pyridoxal phosphate, and this holds that amine group, which is then transferred to another alpha-ketone acid, and a new amino acid is made. And this is done within our body to create new nitrogen products, or to be held within a nitrogen pool by glutamate to form new amino acids later on, that your body can synthesize these non-essential amino acids. The other pathway that can happen is your body can go through oxidative deamination. And in a healthy person, your body should be going through, your liver should be going through this 10 to 15% of the time. This is the liberation of the free ammonia from the amino group coupled with oxidation. So this provides a way for the ammonia, NH3, to become NH4 ammonium and go into the urea cycle, and then the carbon skeleton that is left over will be used for energy. So here we have oxidative deamination. You have this glutamate right here, which is the nitrogen holder, the amine group holder, where the ammonia is going to be taken off of the glutamate and become alpha-ketoglutarate, and this ammonia is going to become urea eventually. It's going to be converted into ammonia and then enter the mitochondria of the liver, and then through the addition of ATP, CO2, and bicarb is going to enter the urea cycle and become urea. This will then go to the kidney to be excreted through the urine, because ammonia, the NH3, is toxic within our body, and we want to be able to get rid of it. And this urea cycle is a way for our body to get rid of that through the urine. But what happens if the oxidative deamination is going on too much? So when oxidative deamination is happening more than 10 to 15% of the time, your ammonia ends up spilling into the blood, and it ends up making its way to the brain and crossing the blood-brain barrier. Your brain doesn't have the urea cycle happening. So what happens to compensate for this, there's an increase in the glutamate and glutamine cycle, where when you have an increase in the glutamine within your brain, this causes swelling and edema because this attracts water. Interestingly, also, with an increase in glutamate, this represses GABA, which is inhibitory, and glutamate is excitatory, which is going to cause excitotoxicity when you're going to have an increase in anxiety. Also, with people who are metabolically inflexible and are completely reliant on carbohydrates or sugar, are going to fall into a category where their going into sarcopenia, where they're muscle wasting. They're actually using their muscle and eating away their muscle to provide energy. They're going to be going through oxidative deamination so that they can get rid of the amine group and then use the carbon skeleton that's left over as an energy source to get glucose. This is not good because this is essentially like using throwing, if you were to say, if you had a fire at your house in the fireplace and you were taking your own furniture and putting that into the fire to feed and to flame your fire. That's not very efficient, and it's very costly. And protein metabolism is very costly. It's energy costly, so that is not what you want to be happening. So thank you for watching this video.